So I want to introduce our guest today, Dr. Hussaini. He is a neurobiologist who conducts research regarding Alzheimer's disease and brain aging. He is also currently a professor at Columbia University, where his lab is working with rodents to identify neurons that are not functioning well and or correctly during the early stages of Alzheimer's, and is looking at ways to block the progression of the disease. So he is here to explain to you what all of this means and to answer your questions about being a neuroscientist. So I'm going to stop sharing my screen. And then Dr. Hussaini, you should be able to share your screen now.
Yes. Hello, everyone. Before I share my screen, I want to say thank you to Ellie for having me here. It's a great honor. I love talking to kids. I often entertain them in my lab every summer. Unfortunately, not this year. But I like having, we are allowed to have kids after, I think, the age of 14. So there are lots of kids who come for the first time and try to fumble but learn eventually, but which is fun experience for me because normally I don't teach. I do a lot of research, but I would love to be more interactive with kids. So let me share my screen and I'll go over my slides. Can you see me all right?
Yeah.
Okay, great. So hello everyone again. So I'm going to use my laser pointer so you can see me better. So I'm going to talk on about how Alzheimer's disease affects the brain. Ellie has already outlined what dementia means, that it causes memory problems and stuff like that. But she said maybe I should also talk a little bit about myself. So I put in some slides about myself. So I'll talk about myself, Alzheimer's disease, and then end with my research and what the future of my lab is. And to start with, I did my master's in toxicology, and the first experience for me was to work in actually a forensic lab, toxicology, and we used to see lots of cases where people came with snake bites and stuff like that, and you could see how these toxins alter the state of mind, so which is like very fascinating, and that's how I got into the science of brain. And then I went on to do my PhD on, in fact, honeybee brain. And if you remember, I mean, the way the bees collect honey is they go to a patch of flowers, bring honey and bring nectar back to their hives and deposit to make honey, right? So how do you think they remember where to go and how to come back? So all that little brain remembers how to get back from the patch of flowers back to the hive. So I was studying in my PhD how bees do this, and it was a lot of fun. But then I also wanted to study mice, which are like higher up in the evolution. I specifically studied these smart mice and how they could do a certain part of a maze much better than other mice. So that was my postdoctoral work in Columbia University, and after which it made me realize, if the brain can do really smart things, what could happen if something in the brain breaks? Like in Alzheimer's disease, right? So in Alzheimer's disease, the neurons something is happening to the neurons, which I'll come to that soon. But I was like super fascinated on why this is a problem. Why do people forget was my main question. So I will start here and I'll outline 4 main things on Alzheimer's disease. What is Alzheimer's disease? How it is caused? Whom does it affect, and when does it affect, and what region of the brain? I'm sure Ellie explained to you how the brain anatomy is, so you will get to that towards the end of this talk. So let me start with what Alzheimer's disease is. So if you Google Alzheimer's disease, this is the picture you get. So this is a very nice picture. Because it shows an old woman here who I guess is talking to her family, but she doesn't know whom she's talking to. And this other person is trying to show, oh, this might be one of your grandchild. So and she's trying to ponder who that person is. So it truly captures what forgetting is. So, and this is like part of memory loss. So that's what Alzheimer's disease does. So in the next slide, you will see a movie, Still Alice, not the entire movie, of course. It's a small clip, and it shows a point where her husband is asking, let's go for a run. And before going for a run, she wants to take a quick toilet break. And you'll watch this.
Yes, we are. I just have to go pee. We're right back.
So there, you saw that she went down to look for the toilet, but she could not. So that's one of the first symptoms of Alzheimer's disease. So people usually get lost in their own homes, and this was truly captured in the way she was trying to look, for the bathroom here. So some of the other symptoms are, of course, there are many there. So these are the 10 outlined by Alzheimer's website. And the one to really remember for this talk is confusion with time and place. So that's one of the biggest symptoms and one of the first ones. So if you see any of your loved ones getting lost in time and place, that's like one of the first signs, and you should try and see if you can get help for them, right? So back to some really history of what is Alzheimer's disease, right? So this is 1901 when Dr. Alzheimer had this very interesting patient called Auguste. So she came with a lot of like hopelessness, then that she was trying to forget the things, even including her names. And he noted that she had like a lot of problems, including being very forgetful. And after she passed away, when he looked into her brain, he found something called plaques and tangles. So try to remember these two words because it will come up a lot in my talk later on. So that's how Alzheimer's disease was born, through this patient who had these plaques and tangles in her brain. So Ellie probably outlined how a brain looks like, and you would remember this is a neuron, and it has these dendrites and axons, and this is a normal brain. But when you look at the Alzheimer's disease brain, it is shrunk considerably, and you see how much smaller it is. And something you will note is within the neurons there are these tangles, and there are plaques in between the neurons. So this little video will outline what I'm talking about, right?
Neurons constantly communicate with each other through electrical charges that travel down axons, causing the release of chemicals across tiny gaps to neighboring neurons. Other cells in the brain, such as astrocytes and microglia, clear away debris and help keep neurons healthy. In a person with Alzheimer's disease, the most basic form of dementia, toxic changes in the brain destroy this healthy balance. These changes may occur years, even decades, before the first signs of dementia. Researchers believe that this process involves two proteins called beta amyloid and tau, which somehow become toxic to the brain. It appears that abnormal tau accumulates, eventually forming tangles inside neurons. And beta amyloid clumps into plaques, which slowly build up between neurons. As the level of amyloid reaches a tipping point, there is a rapid spread of tau throughout the brain. But tau and beta amyloid may not be the only factors involved in Alzheimer's. Other changes that affect the brain may also play a role over time. The vascular system may fail to deliver sufficient blood and nutrients to the brain. The brain may lack the glucose needed to power its activity. Chronic inflammation sets in as microglial cells fail to clear away debris, and astrocytes react to distressed microglia. Eventually, neurons lose their ability to communicate. As neurons die, the brain shrinks, beginning in the hippocampus, a part of the brain important to learning and memory. People may begin to experience memory loss, impaired decision-making, and language problems. As more neurons die throughout the brain, a person with Alzheimer's gradually loses the ability to think, remember, make decisions, and function independently.
There. So that was like the main gist of what Alzheimer's disease is. And you saw that they explained amyloid beta and tau protein. So these two proteins have normal function, but they turn bad. I mean, once they turn bad, they become plaques and tangles, and that's what happens. So plaques mainly affect the hippocampus early on. Later, it dominates the entire brain. Same with tangles. It starts with entorhinal cortex and then moves over to the entire brain. So I'll come to that later. So in general, who is Alzheimer's disease affecting? So there are like two kinds basically. One is late onset and early onset. In the late onset, it usually is affecting 65 years and older people, like 1 in 10 people. So this is the most common. This is very rare. And among the late onset, women get it more than men. And one of the gene that is implicated is ApoE4. And this is like, and anybody can get tested, right? You can go now to the doctor and get a genetic testing and make sure if you have like like 2 copies of this gene, you can maybe take some precautions early on to offset the changes, right? The early onset is different, which is what you saw in the movie clip I just showed you. Still Alice, it's a really rare kind of Alzheimer's disease, like only 10% of overall Alzheimer's disease have these, and it's really early in like 30 to 60 years, it appears. And these are the 2 genes which are implicated with the early onset. And again, you can get yourself tested to make sure there are no like mutations in your gene. So those are the main people affecting, and those are the ages, right? So from all this, I mean, we can like narrow it down to one region where it appears, right? This is called the temporal lobe because it's around your temples, basically. That's why it's called the temporal lobe. And then you know that that lobe has 2 main regions, that is entorhinal cortex and hippocampus. Incidentally, they are both very important for learning and memory. So now I don't work on humans, but I do work on mice, which also have these 2 regions, hippocampus and entorhinal cortex. And the reason I work with mice is they're really easy to work with. They can quickly generate in like short time. You can have like artificially inject them with tau and A beta, the plaques and tangles I was talking about. So we can like study them indirectly that way. So that's what my lab does. We study plaques and tangles in entorhinal cortex and the hippocampus, right? So I'll switch gears and go to a completely different question, but it's related, right? So you all know what a GPS is. So we have a map on a GPS, and it basically it's helping you go to a specific location. So a GPS needs a map on it. It needs a location for you to go to, and you need a direction to know which direction you're going. So interestingly, our brain has a GPS. So our brain's GPS is actually the hippocampus, the entorhinal cortex, and the subiculum, but we won't like focus on the center part today. But I'm going to study this blue region and the violet region in my lab. So what we do is we use like really thin wires. So these wires are picking up electricity. So you might have studied that neurons give out electricity, tiny amounts, and we can use almost like these microphones to pick up electricity like this from the neurons, and that signal can be used to understand how the brain works, right? So what I'm doing is now I'm sticking electrodes either in a hippocampus or entorhinal cortex, and we can study the GPS neurons, right? So on your left is a recording from hippocampus, and you can see the animal running around here, and the red regions here indicate where the neuron is active or firing, right? So you'll see a pattern emerge, and you'll see over time there is something special about this place. So once the video ends, you will see, yeah. So that's like a really active region for that particular neuron, right? Now I will show you another neuron, but this one now is from entorhinal cortex, and you will see a mouse walking around, and slowly there is a pattern that is going to emerge. And you will initially won't see it, but as the animal goes faster and faster, you will see like an interesting pattern emerge, and I'll let you figure out. Right, do you see there is like a grid-like pattern? So they're like, so this is like one single neuron in the brain, which is making this beautiful grid-like pattern. So what is happening in entorhinal cortex is there is this neuron called the grid neuron, and the place you saw was the place neuron where the animal was running. There's like one place, and this grid neuron was one. So now we want to understand if this, what happens to this when you put plaques in. So when I put my plaques into a hippocampus, what would happen to the place cells? So this is what happens. So when there are no plaques, our place cells are perfectly firing, our neurons are firing perfectly well, but when there are like plaques, you see the neurons are affected, and the firing region has changed a lot, right? And similarly now with, if I put my tangles in entorhinal cortex, then you see in a normal case, it's a nice pattern, a grid-like pattern, but if you have tangles in there, your pattern is lost. So it's trying to tell us that the plaques and tangles are causing the neurons to dysfunction. So that's what it means, right? So not only does it affect the neurons, we know that neurons affect behavior. So so we use like three very common behaviors in mice. One is the water maze. In water maze, you see this little blue region. We put the animal, that's a platform for the animal to climb on, and the rest is all like water. It's almost like an island. So the animal is, the mouse is put here, and it has to learn to swim and find the platform. So after some time, it goes directly to the platform because it has learned. But if with the animal that cannot find the platform means it doesn't remember where the platform is, it doesn't have the memory for it. So second behavior is the object recognition. So if I show you these two, you will immediately point out that, oh, there's a triangle here among a rectangle, right? But a mouse which cannot identify the new object also has memory issues. So we test this all the time. And with the T maze, because it's a T-shaped maze, we give food reward on this side or the other side, and the animal has to remember which side the food is. And if you cannot remember the location, it's supposed to be impaired in its behavior. So these are the three main behaviors we use to test if the neurons are affected and the behavior is impacted. And like Ellie pointed out, in dementia, there is no cure. So the only ways people are trying now is to improve memory with medicines. And the other research that is ongoing is trying to identify disease very early on and trying to reduce plaques and tangles in the brain. So regarding this last point, we had some success in my lab. What we are trying is in the mouse brain, remember there's a left side and the right side. And I showed you before that there are like plaques and tangles deposited. What we did was only on the right side, we tried to reduce the brain activity, and we see that only on the right side, there was a reduction in plaques and reduction in tangles. So you see, it's much lighter on the right side. So we were able to achieve reduction in plaques, but we don't know if the behavior was improved. So that's my future, right? We are trying to identify plaques and tangles earlier, trying to identify if there are memory problems that are earlier, and also improve memory. So one of the things we are doing in the lab is, I hope this time I can manage. So you'll see this animal is implanted with a blue light. What this does is it's like a really cool neuroscience tool where you can control the neuron activity just by shining light. So when I do this, you see this thing is like flickering the light. So what it is doing is we are trying to control the activity of the animal's brain and try and hopefully improve memory. This is what we are trying for future. So we don't know if it's going to work, but for now, the other thing we are doing is we have these new virtual reality environments for animals. So the idea is we want to be able to be recording from many, many neurons. So because the mouse is really small, so we have, and has to carry many electrodes, so we have tried to fix them in one place. Let me pick this. Yeah, we are fixing the animals in one place and as they're playing video game, we're trying to record from their brains. Yeah. So thank you very much. I hope you enjoyed the lecture. Thank you.
All right, so now I think if anyone has any questions, you can feel free to ask or type them into the Zoom chat box so that Dr. Hussaini can answer your questions. I have a few, if anyone's shy, that people have already submitted.
Sure, go ahead. Happy to answer.
Okay. What is it like to study the brain in the lab? I think this was Julia's question.
Okay, so because we are using animals, it's very easy because there is like a less complication with humans. There are like too many, you have to worry about the person's family and stuff like that. With animals, there are so many controls in place, but still we are able to study systematically what is wrong. Like I said, we are able to genetically change the animal's brain to have Alzheimer's disease, and we can do a lot of fine tuning to study the brain compared to what we can do with humans.
All right, Jasmine asked, how do you get mice to play video games?
Good question. So it takes them a week to play video games. All we do is initially they don't understand that you have to run for the screen to move. But I think we also like tempt them with treats. And once we keep doing that, they automatically learn. So what I didn't show you in the video game, that they have to go to some places to get a reward. So once they reach like a certain room, they receive like a sugar water or like a treat. So they learn to do that pretty quickly.
Right, here are a couple other ones. So, how are you able to put the tiny wires into the mice's brains, and also how you are able to put the plaques and tangles into them as well?
Yeah, so we have to do like how neurosurgeons implant electrodes into humans. So we do these surgeries in the lab where we can implant these tiny electrodes, which are like less than a size of your hair, half the size, I would say. And once they're inside, they are good for recording. And for plaques and tangles, we have two approaches. One is genetic approach, where you can already buy, like you buy your pets from a pet store. We have like these big companies who sell these mice which already have plaques and tangles. The other way is, somebody asked this question earlier. So we have lots of donors who donate their brains. So we can use tangles and plaques from their brains and inject into a mouse brain, and then we can study them directly. So actually, it's pretty cool to study human tangles into a mouse brain. Yeah.
Right, does anyone have any other questions?
Somebody asked, where do mice come from? I think, yeah, they come from certain companies. If you're interested, I can tell you. Ben, thank you.
Right, someone else ahead of time had asked something. What is your favorite part of your work?
Oh, I think definitely behavior with mice. So they are really interesting. Although they all look the same, each mouse has a personality. And if you work with them long enough, you will understand that each one of them has like a quirk or an interesting habit. So even though we test like so many mice at a time, they all have like different habits, and the behavior is the best part. You get to do so many mazes. They have to navigate like a tea maze or do this water maze and stuff like that. That's a lot of fun.
Ellie, I had a question.
Oh, sure.
Hi. So you had mentioned that there were tests that you can have done early on to measure for late typical dementia, Alzheimer's or early onset.
Yeah.
Is that really recommended for family members who have family members who have had Alzheimer's or been diagnosed, or is that something that you're finding more and more people who are doing?
So yeah, I know people who have Alzheimer running in the family, and especially if you had the early onset one, if they got Alzheimer much before 65 years, then there's a likelihood that there is a small chance it runs with the family, right? So, but it might not be the case. So our neurologists in Colombia, they are routinely suggesting people to get their genetic testing done to make sure to eliminate one fact of it. If it is a lifestyle change, that's a lot easier. With the genetic, you have to just live with it, I guess.
Right. So I was going to say, if they did find that they had that gene, then they could take steps to be able to help with that.
Yes, yeah, because especially the ApoE4 I mentioned, it's a very rare one, but if you do have it, there are some steps one can take in terms of like lifestyle changes. I think it's good to know beforehand if you have those genes early on. Otherwise, I don't think you should worry too much.
No, it's just good to know. I find this very interesting. Thank you so much.
Sure.
Yeah, so what makes Alzheimer's different from other brain aging related diseases?
Okay, so that's a good question. So with brain aging, the process of forgetting is really, really slow. It happens very gradual. But with Alzheimer's disease, the problem is, I mentioned two bad proteins, the plaques and the tangles. The intersection of them is the most important part. And when that happens, it goes like within 5 years, a person can be really normal to completely forgetting who your son is, for example. So it like hits really fast when it happens. So that's the most scary part of Alzheimer's disease. Yeah.
Right. Does anyone else have any questions for Dr. Hussaini before we have to end the session? No? All right. Well, thank you so much for joining us today for both of our sessions today. I know all the kids were really interested in learning from a professional neuroscientist. So
thank you very much. Yeah. The one thing to add is feel free to ask me more questions. You can share my email with them if they want to ask me something in a separate way. So happy to answer. Thank you.
yeah, thank you so much. All right. So before we end our session, let me share my screen real quick. Yeah. So as always, continue to keep checking your emails for information on next week's session. I'll be sending that information out tomorrow. Remember to keep working on those end of series projects, right? I've gotten some really, really great feedback about some of them and some questions. So if you ever have any questions, keep emailing me about those. Make sure you're also submitting those to me at least a day or two before next week's session so I can compile them and they're all good to go so we can present them next week. For all of this information that we have gone through today and Dr. Hussaini's really interesting videos of those mice in his labs, you can always go back and look at that once I post them on our website, thebrainofthebrain.com. That'll probably be done sometime tomorrow. So you can definitely check that out and look at that. So that is all for today's session. Thank you guys for coming and I'll see you next week.
Okay, see you guys.
Bye.
Bye-bye.